Good evening, ma'am.
Good evening, sir.
Good evening. Good evening, sir.
Good evening, Pa. Good evening, Dhanalakshmi.
Sir, you can start now, sir.
Yes, ma'am. Thank you. Good evening, my dear students.
Good evening, sir.
Good evening. Once again, you are all are warm welcome to the biology classes. Already we know earlier about what are the structure of RNAs. Already we studied, isn't it? In the last class, okay, the tRNA, how many types of arm is there? And there are 5 arms is also there. The next is mRNA. mRNA, how it will be comes from, what they are carrying. Okay, all those things already we discussed, isn't it? So this is considered as a nucleus. You know about these slides we are showing. Nucleus is there. Inside there is a DNA molecule. The DNA molecules undergo the transcription process as a result of the formation of mRNA. The mRNA after the formation it will enter into the nucleus into cytoplasm through the nucleophore. Okay, so that things already we discussed. And then, so what are the coefficients? mRNA sedimentation coefficient 8s. How many nucleotide is there? How many minutes it will take, or otherwise called as a lifespan of mRNA is how much in prokaryote, in eukaryote, that things also already we discussed. What is by introns? Introns is nothing but the non-coding sequences also present in the, which one you know, eukaryote, it's completely absent in the prokaryote mRNA, that things also already we discussed. And approximately how many molecules, how many nucleotide it is made up of, that means 900 to 1500 nucleotides. It's made up of mRNA. And also it is also divided into 2 according to their structure. One is called as monocystonic, okay, in eukaryote, and polycystonic in bacteria and phages also already discussed. Okay, and then, so what is my untranslated region? UTR, this is the untranslated regions, which is the starting codon. This is the protein coding region, and also this is the stopping codon, and also opposite side there is a 3' end is also there. The 3' end also it has a untranslated regions. So we come to the conclusion both the side 5' end as well as the 3' ends, there is an untranslated region is also there. And 5' end is the starting codon. Starting codon is started from where, you know, in the 5' end only. And also the process will be continuous, already you know about. So this is the entire structure of the eukaryotic mRNA. So this one is, what is that? A bacterial mRNA, otherwise called as a prokaryotic mRNA. Okay, this is, what is that? Prokaryotic, because bacteria is also comes under the headings prokaryote. Okay, see that one? So this is the eukaryotic mRNA. Eukaryotic mRNA. Okay, so already we discussed. See that one? The prokaryotic mRNA polycystonic. What is the meaning of polycystonic? Means this is considered as a more than one protein synthesis phase is there, otherwise called as starting sites also. Say this is the light blue color is the starting stage. So each one it has a specific protein molecule biosynthesis take place. That's why it is considered as a polycystonic. See that here one, here one, here one. Cystron is nothing but the small segment of the DNA nucleotide responsible for the protein biosynthesis. It carries the codon, and also it essential for the synthesis of protein molecules. Okay, here how many is the different color? Green and violet or blue in color. So the here the 3 protein sequences, otherwise called as translated in short sites, also present in it. So that's why it is called as polycystonic, mostly present in prokaryote. Prokaryote polycystonic, eukaryote monocystonic. So here the only one protein site only present in it. That is the reason. Okay, that things already discussed. Next, we are going to discuss remaining part of the tRNA molecules and the mRNA molecules. The mRNA molecules, there are three primary regions also present in it. Don't forget, my dear student. This is very, very important. Already we saw it, in the previous slides. There are three primary regions of the mature bacterial mRNA. Okay. So the first one is 5' untranslated region. Second one is the protein coding region. And the third one is untranslated region. What is that? 3' untranslated region. Already we saw it in the previous slide. The sign Dalgarno consensus sequence. What is that meaning? The sign Dalgarno's consensus sequence in mRNA is required for the attachment of the small subunit of the ribosome. This is very, very important. You know about the protein factory. Which one is called as protein factory? Which one is called as protein factory, my dear students?
The ribosome, sir.
Okay, because it consists of two components, isn't it? What are they? The upper part, there is a large subunit, and lower part, there is a small subunit. Okay. So in between, there is a mRNA is there. Okay. Already we saw it in the previous slide or class. That sequences is called as what is it? Sign Dalgarno consensus sequence. Okay. It is help for the attachment of the small subunit ribosome. Small subunit means what is that? mRNA attached with the small subunit of the ribosome. Which one is helping here? Sign Dalgarno consensus. See the next slide, you can easily understand. See that? So this is the, in prokaryote mRNA, the untranslated region is called the sign Dalgarno sequence. Okay, what is that? The prokaryote or bacteria is an example. It has mRNA, okay. The mRNA, that untranslated region. Untranslated means what is the meaning? It cannot be able to mention or expose any amino acid. That region is called a sign Dalgarno sequences. So this is the structure. See that one? -10 sequences is that. -10 already we discussed, isn't it? -1 to 10. Okay. See that? The 5' to 3' N. This structure is called as what is it? Sign Dalgarno sequences. See here, AGG, AGG, U. The starting is what is that? 5' N, AGG started. AGG again, double time it's repeated. And U, 3'. So this structure is called as what is that? Sign Dalgarno sequence. What is the main use of this one? This is used for the attachment of the mRNA with ribosomal RNA. See the structures. See that one? This is the mRNA. Okay. So structure is 5'. Here already know about 3' in the opposite side. AGG, AGG, U. 5 to 10 nucleotides. Okay. AUG is a starting codon. Already know about this is the protein biosynthesis starting codon. Okay. Corresponding combination, how it will be joined. A always combined with the molecules of uracil, guanine, C, cytosine. Again, guanine, cytosine, adenine, uracil, guanine, cytosine, guanine, cytosine. Uracil, you know very about the adenines. This structure is called a 16S or RNA. So this entire structure collectively called as what is sign Dalgarno sequences. It is essential for the protein synthesis initiation or starting point, like that. AUG is the starting codon, isn't it? Before that one, we need which one, sign Dalgarno sequence, and only it will be proceeded. See the next one. It is purine rich and is part of the ribosome binding sites because it's containing purine molecules highly. What is my purine? adenine, guanine only. Okay, these are the purine molecules. And highly rich in this portion, what happened? It can be easily bind with the ribosomes. So that part is called as what is ribosome binding site. Okay, so this is very, very important. And next one is it is complementary to the ASD, anti, what is that? SD sequence. Okay, anti-SD sequence, that is denoted as ASD. Present on the 3' end of the 16S rRNA of 30th S subunit or ribosomes. Already know, but there are 2 subunits. Already know. Yes. Large subunit is also there. Right. Okay. The lower part, there is a small subunit also there. Yes. Okay. When the Sini-Dalgarno sequences attached with the 3' end, okay, 3' and 5', this is the combination, isn't it? In the ribosomes, what happened, see here, the complementary of the ASD, anti-SD sequence. So it is present in the 30th subunit. That is very, very important. And mRNA also has some additional sequences that are not translated and are referred to as the untranslated region. That things also already we discussed in the slide. See that one? So this is considered as an untranslated region. This is considered as untranslated. See, this is the untranslated regions. This is also untranslated regions. Okay, see that one? Okay, the untranslated regions are present at both the 5' end before start codon and the 3' end after the stop codon. Here the spelling mistake. So this is the important things. Already we saw it in the previous slide also. Once again, repeat for you. The mRNA as well as the Sini-Dalgarno sequences also, it has an untranslated region in both the side, 5' end as well as the 3' end. In between, there is a, what is that? Protein biosynthesis codon is also present in it. See that one? This part. See, translational codon. This is translation control, subcellular location, stability. This is the structure, exact structure of the mRNA. See that one? The 5' end untranslated. UTR is denoted as untranslated regions. Mostly they ask the question in which one you know? Explanation or mnemonics. This is the mnemonics. UTR is nothing but the mnemonics, short form. Okay. So this is the 3' UTR and 5' UTR. In between coding regions, that is even here. Okay, coding regions. How it will be. Okay, so these are the structures. They are required for the efficient translation process and provide stability. What is the main use of these? Suppose if it is not bind with the ribosome, what happens on the protein synthesis, otherwise called as translation process, it will not occur. So the stability maintained by the, which one this component, then only it will be proceed. Otherwise, what happens? It will not proceed. Usually, each gene transcribes its own mRNA. Usually, each gene transcribes. Each gene means what is that? The DNA present inside the nucleus. That gene transcribe that own mRNA. That is the reasons. For example, if you want to biosynthesis of the, okay, and heparin protein, so it has a specific transcription of mRNA. Likewise, globulin, keratin, immunoglobulin, albumin, okay, other enzymes also. Each one it has a specific transcripts its own mRNA only. There is a message. Therefore, there are approximately as many types of mRNA as there are genes. Already told you. Okay, in cytoplasm, there are so many mRNA molecules present in it. Each one it carries their specific amino acids which joined with the which it is also carried from where cytoplasm into ribosome, which one is matched to the anticodon. Okay, that is very, very important. Because of the variation in the size of the mRNA population in a cell, The mRNA is often called heterogeneous nuclear RNA or hnRNA. So sometimes they may ask the question, what is by heterogeneous nuclear RNA? Sometimes, otherwise hnRNA. What is the expansion of hnRNA? Okay, like that they may ask the question. You have to answer it properly. It is nothing but the heterogeneous nuclear RNA. Which one of the following statement is incorrect? Which one of the following statement is correct? Like that they may ask question. Okay, according to the various size of the mRNA population, it is called as what is a heterogeneous nuclear RNA. They are having the same. Hetero means what already in our, but it is nothing but the different. Okay, heterozygous. What is the meaning of heterozygous? In male, there is a heterozygous because already know about, isn't it? What is the meaning of male? See that one? Take an example. Some male gamete is there. So, and female gamete is also there, isn't it? Female gamete is also there. Male gamete already know, but there's nothing but the sperm. Okay, nothing but the sperm. Female gamete, just take an example of egg. These are all are what I mean, according to their size and shape, they may be vary. That's why they are all are comes under the heading known as what is that? Hetero gametes, isn't it? Gametes. Got it? This one is heterogamous. Suppose if it is both are same, that is called as what is that? Both are same in nature. What is that? This is called as isogametes, otherwise called as homogametes. Okay, so that is very, very important, my dear student. Okay. Say that one. So heterogeneous nuclear RNA, the mRNA is also called as what is that? Genus rRNA because the size is vary. Okay, don't forget, my dear students. Go to the next ones. When one gene, the one gene is called as cistron, particularly the one gene is called as cistron code for the single mRNA molecules. What is by cistron? Cistron is nothing but the only one gene present in the mRNA molecule, that is called as what is that? Cistron. Suppose if it is more than one gene, it is called as polycistron, otherwise called as polygenic, otherwise it's mostly called as prokaryote. Already we saw it in the previous slide. Okay, see this one. So this is the structure. Okay, this is the structure. Okay, so it can be placed here also. Okay, so this is very, very important. See this part. You have to place it here also. This part. See, prokaryote, it contain what is that? So many cistron. These are all are the protein 1, protein 2, protein 3. These are all are cistrons only. But what about here? They only one cistron. That's why it is called as monocistron. This is called as polycistron. That is mentioned here also. Okay. Okay. The polycistronic mRNA has an initiator codon. Already nothing but the starting codon. Don't forget, my dear. Polycistronic mostly present in the prokaryote. Already told you. Example is bacteria, otherwise called as lower organisms such as the bacteria as well as the bacteriophage also, otherwise called as virus. The polycistronic mRNA has an initiator codon. Initiator codon is nothing but the starting codon, that is mentioned as a AUG or GUG. Don't forget, my dear. This is only in the polycistronic nature. Suppose if it is the monocistronic, take an examples of higher animals. AUG is the starting codon, already you know about. And the terminator codons, take an examples. See UAA, UGA, and UAG. So these are the three codons. So starting codon is 2. They have a mention. AUG is a starting codon. GUG is also a starting codon. Stop codon means what is that? UAA. Just don't forget, my dear student, this is very, very important. UAA and UGA or UAG. These three codons, when it comes to the end of the reaction, what happened? You know, the protein biosynthesis, otherwise called as the polypeptide chain completely stop. For each polypeptide to be formed by it. Okay, without these two things, there is no any protein biosynthesis, otherwise called as a translation process. It will not occur. That is very, very important, my dear student. Which one is starting codon? AUG and GUG. Which one is the stop codons? What are the stop codons? Is the UAA, UGA, UAG. It's just you take these three. Okay, just take the three. All are, see here, A, U, U, U. AA, GA, AG. Okay, you can shuffle. You put here UAG, UAA, UAA, UAG, UGA, like that, for your convenience. Which one it can be easily rememberable. Okay, so that is also very, very important. So without these things, it will not proceed. Okay, so already we saw it. These things, protein molecule is there. So this one is untranslated region. This one is also untranslated. Starting codon is also there. Here the AEG starter for this protein molecule. Here is also AEG, the protein molecule. Okay, so this is the important. Although the tRNA are quite stable, tRNA are quite stable in prokaryote, they are somewhat less stable in eukaryote. This is very, very important, my dear students. Sometimes it may be asked the questions. What is that? The tRNA molecules quite stable in prokaryote. They are somewhat less stable in eukaryote. This is another important point. Okay. The opposite side is true for mRNAs, which are quite unstable in prokaryote, but generally stable in eukaryote organisms. What is meant by opposite and true for mRNA? This is denoted as mRNA opposite, means this is considered as a, what is that? Anticodon. Okay, opposite of the, which one, mRNA is called as, what is that? Anticodon. Anticodon, how it will be, which are quite unstable in nature in prokaryote, but at the same time generally stable in the eukaryotic organisms. Okay, this is very, very important, my dear students. Don't forget in this part here, you must note it down. What is meant by polycystonic? What is meant by monocystonic? What is meant by sign Dalgarno sequences? How many sequences present in it? How it will be started? Starting from the 5'N to 3'N, AGG, AGG, and also U. Okay, so another one is, what is that? It is denoted as the next one is called as, what is that? Polycystonic and monocystonic nature. Okay. So next we will pass on to the next one is question part. You have to answer it, my dear students. So already you studied these things. Will you please give me the answer for this one? Any one of you? Width of the DNA molecule is very good. Ajay, sir. Yeah.
Sir, it's option C, sir. 20 angstrom.
Very good. Exactly correct. Okay, 20 angstrom. Which type of DNA these molecules, my dear? These molecules. What are the types of DNA molecule present in, my dear? Different types of DNA molecules. Will you please? A, B, Z, isn't it? D also. So this is comes under yeah. You must note it down, my dear. Okay, it's very, very important. Types of DNA molecules according to the structure and also the width also. Not only the width, and also number of nucleotide present in between the rotation. Each and every rotation, it has a 10. Some of them are having 8. Some of them are 12. Some of them are having 10 also. Okay, 11, 12, like that. And also in between there is a structure also reduced. Okay, anyhow, right. Very good, Ajay. Keep it up. Okay, what about the next one? What about next one? See that one? You can easily identify. Which of the following RNA is most abundant in animal cells?
Sir.
Now only we studied. Animal cell, how many percentage most abundant? Whether it's mRNA, whether it's a tRNA, whether it's a rRNA, whether it's a small one, micro RNA.
Sir, it's rRNA, right, sir?
Yes, already know about 80% already we discussed, isn't it? Is it
Yes,
correct
sir.
or not? Is it correct or not?
Sir, it's rRNA only, sir. Option C, sir.
rRNA only. Are you sure, Ajay?
Sir, yes, sir. Sir, that's cause mRNA is 3 to 5%, sir, and tRNA is 15%, 10 to 15%, sir. So it's rRNA, sir.
Okay, suppose if it is wrong means, suppose I will confuse you. Don't forget. Okay, give your answer confidently.
Yes, sir.
That is very, very
rRNA only, sir.
Yes, we will see last. Okay, what about Dhanalakshmi? Please answer me, my dear. Okay.
Yes, sir.
Okay, go to the next one. A one gene, one enzyme relationship was established for the first time in which type of Neurospora crassa, Salmonella typhimurium, E. coli, and Diplococcus pneumoniae. Which is the correct one? One gene, one enzyme relationship was established. See the structure.
Sir,
See the structure. You can already we saw it this one so many times. One gene, one enzyme hypothesis was discovered by Pidilantatum by the fungus is called as fungus. Already told you. Which one is fungus here?
sir, Neurospora crassa.
Very good. Neurospora crassa is a fungus. Others are Salmonella typhimurium, E. coli, Diplococcus. All are considered bacteria only. So this is the correct one. Okay. Go to the next one. Virus that infect bacteria multiply can cause their lysis or called. It is also one of the life cycle of the bacteria. Bacteriophage after the which cycle this one? Lysozyme, lipolytic, lytic, lysogenic. Which is correct here? Lysozyme, lipolytic, lytic or lysogenic. In this part, you must know lysosome is nothing but the small cell-bound organelle present in the cell. Lipolytic means lipid molecule lysis only. Here, what happened on the bacteria lysis, isn't it? Lytic means what is that?
Sir,
Lytic
sir,
means
sir,
yeah.
Sir, lytic means it's destroying, sir.
Okay. Sorry, sir.
Sir, yes, sir.
Okay, we will see. Okay, lysogenic means lysogenic. Okay, you are correct. Okay, lytic is the correct one, I think. Okay, we will see the last one. What about Dhanalakshmi? What about your answer?
Sir, lytic only.
Okay, good. Very good. Okay, go to the next one. The process already we discussed this past. The questions already we discussed in the previous lessons also. Okay, see that one. The process 1, 2, and 3 is the image respectively or DNA to DNA replication. Isn't it? Yeah, good. DNA to mRNA transcription.
Sir, transcription, sir. And mRNA to protein, it's translation, sir. So option C, sir.
Okay, see the first one, the answer is C. Which one is the answer? See that one, C. Okay, C. Which one? Is it correct, my dear?
rRNA.
No, here. 20 Armstrong, isn't it? 20 Armstrong is the correct one. Just take a small notes also. And again also, there is a C, rRNA. RNA because already Ajay is correct. Okay, you also correct. Neurospora crassa is the correct one because A is the answer. Last one, C, lytic, C, and CCA, CC is the options. Okay, see that last one is also C. So total answer is C here, capital C, CC, the answer. Third one answer. The first, third question, the first one is the answer, and the remaining, the CC is the answer. So this is the end part of this. Okay, see that one. Okay, my dears. So next, so we'll pass on to the next one is the part 7. Okay, we'll see it. Next we saw it, and we will pass on to the next one is part 7. So in this category, so we are going to discuss about, okay, we are going to discuss about the post-transcriptional processing of RNAs after the transcription. Okay, what are the process it occurs after the transcription? Means mRNA synthesis. After that, what will happen? Okay, it will enter into the protein biosynthesis. That process is called as translation process. Okay. See here, when the transcription of bacterial rRNAs, ribosomal RNAs, and tRNA is completed, they are immediately ready for use in translation process. Before that, what is that? Before translation process, there is a pre-transcription process also occurs. That is nothing but the synthesis of rRNA and the tRNA molecule completely. After the complete productions or synthesis of the mRNA, tRNA and rRNA, what happened? It will enter into the translation process with the mRNA. Okay, this is the first step. No additional processing takes place. You will not take another components. For example, protein biosynthesis translations occurs means it will not synthesize any other, what is that? Metabolic process like that they told. Because within the ribosomes, the translation process only occurs. It will not synthesize any other components, hormones, or any other metabolic like components. They will not say protein. Okay, protein means other proteins. Okay, only one time it will be happen. Only one type of action will be occurs inside the ribosomes. See here, the eukaryotic transmit or proceed in order to the produce the final mRNA. Okay, eukaryotic transcript or proceeds in order to produce the final mRNA. Final mRNA means already you know about the primary mRNA is also there, and after the complete development of the mRNA, this particular codon, this particular protein molecule is synthesized. We need, like that they mention, means that the entire codon is encoded in the mRNA molecules. It has some message, particularly how many types of molecules, how many number of molecules, how many protein molecules they need. That message present in the mRNA molecule, that is complete set of message present in the mRNA molecule. After the complete production of the mRNA from the nucleus, it will enter into the translation process. Before that, what happened? The tRNA and the rRNA also synthesized. So all are, what is that? Ready to participate in the translation process. Okay, so all the three RNAs are ready. So rRNA is also ready, tRNA is also ready, mRNA is also ready. Okay, now they enter into the, what is that? Translation process. Processing of initial transcript includes gapping, polyadenylations, and intron removal splicing. Okay, this is very, very important. Polyadenylation. The first initial step, transcription, included capping and polyadenylation and intron removal. Intra means what is the meaning? In between there is a unwanted codon is there. It also mentioned in between the nucleotide. Completely it will be removed. That process is called as splicing. That is a cutting and removing that process. Okay, so that process is called as splicing. See that one? The ribosomal and the transfer RNAs are also proceeds, but differently. They are either capped nor polyadenylated. So this is the step. So ribosomal and the transfer RNA are also proceeds, but differently. They are neither gapped nor polyadenylated. So next one is gapping of the RNA occurs at the end 5 days end. Note it down, my dear students. So note it down. Gapping. Gapping means what is that? Gapping means, see that one? So polyadenylation. Adenylation, addition of adenyl compound. Okay, polyadenylation, more than one adenylation takes place in the 5 days end only. Opposite side, the introns are removed. That is called a splicing. Okay. After the splicing, what happened? It will be united in the entire chain. Okay. They need, for example, during this process, what happened? And also the molecular scissors also used, and also cut the particular introns and also remove it, and also additions of the remaining compound together with the help of the ligase enzyme. That time, what happened? They need energy component. The energy component is called as triphosphate. Which triphosphate? Guanosine triphosphate. See that one? The gap is a chemically modified molecules of guanosine triphosphate, otherwise called as GDP. Okay, see that one? in a gapping, an usual nucleotide 7-methyl guanosine T or 7-mg. Gapping unusual nucleoside already in our laboratory. We discussed that things also in the previous slide. There is a 5 dash end, the 7-methyl guanine mg they mentioned 7-mg. Okay, the previous slide already we saw it. Otherwise, it will come next slides. Is added to the first nucleotide of the 5 dash end. This is very, very important. That is uridine T or UTR, untranslated region, nearest untranslated region. The methyl guanosine is added. Okay, they may be simply mentioned as a 7-mg. Okay, 7-mg. Which area? The 5 dash end. Okay. The gap help in the proper attachment of the mRNA to ribosomes. The gap help in the proper attachment of mRNA to ribosomes. See the structure. I think here I didn't take a structure. I think splicing. Okay, see here. So this one you can easily see that one. This is considered as a primary transcription. Primary transcription takes place. What is the meaning of primary transcription? Means here, this is the entire nucleotide, right? Here there is a 5 dash end and also 3 dash end is also there, right? Okay. But the first C here, the M7G. What is the meaning of this one? Adenylated compound. Okay. M7G. Okay. In the 5 dash end, there is a gap is there. In between, there is what happened. The nucleotide is also present, followed by exon, intron, exon, intron, exon. It's alternatively arranged. After that, introns exist and exon spliced together. What is the meaning of So all the introns are removed. Here the introns are removed. Here also the introns are removed. The exons are united together here. So you will get the primary transcriptor molecules. Each exons. Each exon it has a what is a nucleotide 1 to 30. Okay, this is exon. And next 31 to 104. So here, what happened? The intron is there. It is not mentioned any codon. It may be removed. So join here 1 to 146 up to this level only. This intron also removed. This one is also removed. Which one is dark one? They may be united together and form as a primary transcript pre-mRNA. Are you understand, my dear? So in this stage, which one is starting one? M7G gap. This is the gap. Okay. This is the stage. Okay. See that one. Yes. This is the structure. 7-methyl guanosines. Okay. Present the 5 dash ends. It is nothing but the gap. So it is used for the attachment of the mRNA to ribosome. Okay. So in between there is a nucleotide is there, already you told exon and intron, exon and intron, exon. Introns are removed, extrons are united together. That complete extron is called as what is primary mRNA, otherwise called as primary transcription, because in between there is a non-coding substance also present, non-coding fragments also present in it. Okay. Suppose it is protein biosynthesis, it will be interfere. So that's why it's completely removed. That position, that fragment is called as intron. So exons are together. It is encoded the amino acid or protein biosynthesis. Okay. So that process is called as what is a primary transcription. Okay. See, the polyadenylations involve the additions of a string of adenine nucleotide to the 3 dash end also. Okay, 5 dash end already we know. 3 dash and also what happened polyadenylations involves the addition of a string of adenine nucleotides. How? See that this polyadenylation is done without the template, the adenine or simply added one after another to the 3 dash end of the RNA molecule. Okay, see that one. The poly A means more than one adenine molecules in the tail. That's why they have given the structure is called as adenine tail. See that one? See this is the structure. This is the structure. See here. This is a normal DNA molecule is there. Here, what happened? There is a gene sequences is arranged in this manner. One is exon, intron, like that. The introns are completely removed. So this part, completely removed. So this part is completely removed. So as a result, you will get these, see the structures. Okay. So this is the 5' this is the 3' So the pre-mRNA, that is called as a primary transcription takes place. That is pre-mRNA formation takes place. After that, it is also spliced into mRNA, complete sequences. These are all encoded the nucleotide. In between, non-coding fragments also present. This is removed. Okay. So we'll get the entire structure of this one. See that one? The structures. This protein 3, you have to move on to that one, I think. Okay, see here. See here, the Mg. Okay, methyl 7 guanosine gap is there. This is very important for the adhesion of this is the last side. See the 3' poly A tail. What is the meaning of tail? It's also extension of that primary or the normal transcription mRNA. Okay, so here what happened? More than one amino acid united in the tail after the 146th codon, triplet codons. So that thing's given here. Okay, see that one. Okay, see that one. Okay, so 3 of the RNA molecule. This poly tail, which average about 200 about nucleotide in length, help in the protecting the RNA from degradation and play other regulatory role. What is the main role of the 3' end or poly tail? 3' end means what is adenine? How many adenine molecules present in the tail? 200 molecules of the nucleotide. Completely. Polyadenine means more than one adenine molecules present in the tail. What is the use of this one? The length help in the protecting the RNA from the degradation and play other regulatory roles. Okay, it is a very, very important points. Okay, one side what happened, adenylation takes place. And the opposites of what happened, 5' polyadenylation takes place. And 3' what happened, what is that? Adenine. Okay, see that one. So that is a polyadenylation is nothing but this one. And previous one already we saw it. What is that? See that one. Methyl guanosine. This is 5' this is 3'. Okay. And some mRNA, including these, for some histone, do not contain polyadenine. Some mRNA, including these, for some histones. What is the meaning of histones? Do not contain polyadenylate. This is very, very important. Another point. Where it present? In the transcription process. Next one is splicing. What is the meaning of splicing? Already you know about. All the 3 classes of RNA are transcribed from the gene that contain introns. In between, there is a introns is there, right? Okay. All the 3 enzymes also formed. 3 RNAs, sorry. 3 RNAs also formed by the removal of exon. Only they contain, which one introns only. Don't forget, my dears. The introns modified into which one tRNA, rRNA, mRNA. They encoded the sequences of amino acids or triplet codon. That is very, very important. Don't forget, my dear. Okay. The sequence encoded by the intronic DNA must be removed from the primary transcript prior of the RNAs being biologically active. Then the intronic DNA molecules, what is that? Completely must be removed from the primary transcript. Then only it will be active. The RNA is active in nature. Okay. After that, what happened? The splicing of RNA is carried out by a large protein RNA complex called a spliceosome. The splicing of RNA is carried out by a large protein RNA complexes. Okay, is called a spliceosome. Entire sequence is completely called as what is a spliceosome. Splicing, each and every is considered as splicing. The splicing pigments of fragments are united together and form as a RNA molecule. Okay, so I will show you here. It will be better for your understanding. Okay, see that one. Take an example. See, So this is the DNA structure. Already we saw it. Okay, this is the DNA structure. Here already you know about. There is a sequences is there. Okay, so this one is called as 5 dash. This one is called as 3 dash. The 5 dash what happened, you know? Methionine. Okay, methyl 7G. That thing's already innumerable. 5 dash N. 3 dash N. What is that? You know, polyadenine. Adenine molecules. Polyadenine means tail. It's nothing but the arrangement like this. Okay, polyadenine tail. In between, there is a sequences there already you know about. First one is called as intron. I denote as intron. This is called as exon. The intron and exon, isn't it? So here the exons are united, isn't it? Exons are coding. These are all are non-coding. Okay, this is the coding molecules, exons. Okay, this one is exons. The exons once united together. Exons are united together. You will get entire structure like this. Exons only united. Okay, so this is the exons. Okay, so this is the part. These are all are only exons. Okay, so this is the 5 dash N as it is. Here you will get 3 dash N's. So this is a structure. This entire structure is called as, what is that? Primary, otherwise called as transcription. Okay, so it is encoded the nucleic acids. Okay, that is very, very important, my dear students. See that one. Okay, see here. Once again, again move to. That is the problem. Okay. See media. Okay. So this one, splicing. Okay, so you will get what is the complete the spliceosome, the entire structure, the primary structure is collectively called as what? No, spliceosome. Spliceosome means more than one RNA complexes united together form as a mRNA. The complex comprises about 150 proteins and it's similar in size to the ribosomes. It is also very, very important. In spliceosome contain how many protein molecules? Sometimes they may ask the question, it is made up of 150 protein molecule is that. Don't forget, my dear. Okay. See that the spliceosome can detect intron exons boundaries, cleave the RNA at the appropriate point and join adjacent exons together to produce a mature mRNA. So primary mRNA, after that what happened? No, the introns, extrons first found the boundaries. Which part, exact part, it may be removed from the RNA. As a result, what happened? All exons are united together and form as a mature mRNA. That is very, very important. We saw it, previous slide that I draw a diagram, that is called as mature mRNA. After the mature mRNA only, they enter into the translation process. Okay. See that condition is called as spliceosomes. The spliceosome are made up of 5 different snRNA pairs. Okay. See here, pronounced SNRPs short for small nuclear ribonucleoprotein. This is called as, okay, SNRPs short for small nuclear ribonucleoproteins. That is called as snRNAPs. Called U1, U2, U4 and U5 and U6. How many molecules there? Here they have given as 5 molecules. See that 1, 2, 3, 4 and 5 only. Which one is here absent? Just imagine. U3 is absent. U1, U2, U4, U5, U6. Okay. So these are the 5 different snRNAP is also present in it. Each snRNP contains one of the class of eukaryotic RNAs, 100 to 200 nucleotides, long known as small nucleus. That is called as, what is that? snRNAs. So this is a snRNAs, right? Okay. How many types, the spliceosome are made up of 5 different snRNAPs. that is called as U1, U2, U4, U5, U6. Okay, these components also present in the eukaryote and prokaryote in different manner, but in eukaryote, what happened, the RNA is 100 to 200 nucleotide long. How many molecules present? 100 to 200 molecules present. It's a small nucleus. Small nucleus means the 100 and 200 nucleotide present in a single polypeptide chain, it is considered as a small nucleus. Suppose if it is present thousands or millions, means it's a long polynucleotide. Okay. So because tRNA, mRNA, and RNA all are, it has a specific, a particular, it is not exceeding the 1,000, 2,000. It's, I think it may be 2,000. Suppose if it is exceeding 2,000, 2 million, 20,000 like that, it is a largest one. Okay. All the tRNAs are this much only. Okay. Don't forget. The splicing signals, how it will be spliced. The most introns start from the sequence guanine and uridine. See that? Starting place is where, the splicing takes place, guanine and uridine. And the end with the sequence A and G, adenine and guanine. In the 5' to 3'. This one is called as 5' and this one is called as 3' and direction. They are referred to as a splice donor and splice acceptor site respectively. Each one it has which one, donor as well as acceptor, then only it exactly bind. Otherwise, what happens? It will not bind with the component. See that one. The U1 SN RNAs containing a sequence complemented to the sequence near the 5' splice site of a nuclear mRNA introns and U1 and SN RNA bind to the region of the primary transcriptions. What they are telling, U1 is the starting point. Already U1 SN RNA containing a sequence complementary. Sequence means there is a nucleotide present in it. Okay, it is near the 5' splice site of nuclear mRNA introns and the U1 SN RNA bind to this region and primary transcription takes place. This is the combination. Which one, U1 sRNA near where is it? The mRNA nearest the 5' splices. So it can be easily bind together and form as a primary transcription takes place. Followed by U1, what happened? U2, U3, U2, U4, U5, U6. Also addition. Okay, SN, RNPs lead to the formation of the spliceosome. So which one is the starting of the splicing signal? The U1. That is very, very important. U1 is the starting point. It is a bind with the mRNA. So as a result, what happened at the starting of the transcription, otherwise called as primary transcription, takes place. Addition with the U2, U4, U5, U6 is also followed or lead the formation of the spliceosomes. Okay, so this is the another thing. The splicing regions start with the cut at the junction of the 5' exons, donor or left, and the introns. Okay. So this is another one. Intron, extron is there. Where there is a cut made by the splicing reaction takes place, the 5' exon. And also the ATP is required for the assembly of the spliceosome. After that, I already told you, exons are united together in the form of, what is a chain-like structure, entire mRNA, isn't it? So that time they need what happened? Energy also. So that's why they provide ATP. The free intron is called as lariat with a sloop and a tail, which are rapidly degraded into the free nucleotide by nucleases. What happened though? The extra things that the intron is there. Intron, what happened? It is not encoded any nucleic acid, any codon. That introns are completely what happened? Degraded by the nucleases. Which nucleases? Endonucleases. Okay, as a result, it will be degraded rapidly. Suddenly, what happened? Automatically, it will be degraded. It is also called as what is that? Lariat with a sloop and a tail. Lariat with a sloop and a tail. Which one? The introns are all called, which one? Free introns. No, don't forget, my dear. Free introns are called as what is a lariat and with a sloop and a tail. Okay, so this is considered here, already know about. See that introns comes out. After that, what happened? It will not unite. The extrons are united with the entire nucleus. So this is a primary transcription takes place. It's a mature mRNA, already know about. So the introns are completely what happened? It it dispose on the cytoplasm and also scattering on the cytoplasm. What happen, automatically it may be degraded by the nucleases. So that process is completely finished. See this one, already we saw it, spliced mRNA. Okay, so the introns are removed. The introns are degraded by the endonucleases. Okay, so this is the structure. The splicing important point. Okay, we will finish it up to this level only today because, okay, so the splicing important points, first described by Philip and Sarpi. Just note it on who discovered splicing. The name of the scientist called as Phillips and Sarpi. The SN RNAs are rich in uridine molecules only, uridine nucleotide, uracil, because it's RNA molecules. RNA mostly found in which one, which one is the highly found, uridine instead of thymine, uridine only present in it. DNA, thymine present in it. In RNA, uridine only present in it because it is also one of the RNA molecule. So it is uridine is a high content. Okay, introns are reminiscent of antiquity, and splicing represent dominance of the RNA world. The introns, what happened, reminiscent and of the antiquity of the splicing represent dominance of the RNA world. These are the 3 points they have mentioned. Okay, so this is the slides. So this diagram is not exactly, I couldn't pick up. Okay, so just to go to and refer the book, you will get you the ideas. So today what we discuss, previous lessons already we know about what is meant by post-transcriptional process in RNA. Okay, what is made translation? How it will be enter the mRNA, polyadenylation. What is the meaning of polyadenylation? And also intron removal is called as splicing. The polyadenylation takes place in the 5' end, and the intron removal also takes place in between the molecules. After that 3 end, what will happen? Already you know about that is called as polyadenylated in 3 end also. The gapping of the RNA occurs at the 5' end. The gap is chemically modified molecules of that is called as guanosine triphosphate. Without GTP, what happened? It will not modified. That the energy requiring process already told you. And a gapping of unusual nucleotide, that is an unusual nucleotide present in the 5' end, that is called as uridine UTR. Okay, so the 7-methyl guanosine. Don't forget, my dear student, this is very, very important. 7-methyl guanine, they may be represented as 7 small m capital G in the 5' end. They can help in the proper attachment of the mRNA to ribosome. What is the main use of this one? The attachment of the mRNA with the ribosome. This is very, very important one. Okay, already you know polyadenylation, how many nucleotide present in it? 200 nucleotide is the. And what about that? What is the main role, about that things also. Others are what happen, completely degraded and also play for the next roles. In splicing, that is introns, extrons are removed. How many nucleotides, how many protein molecules is made up of? The complex comprises about 150 protein molecules and similar in size in your ribosomes. Okay, and also other things also the lysosomes. How many? What is that? 5 types of different types of lysosome all present in U1, U2, U4 and U5 and U6. Nucleotide 100 and 200 because it's a small nucleotides. Okay, splicing signals, how it will becomes starting from AGU and the end with the AG. Okay, so how it will be started already in our report. Okay, so what about the last one? Lariat with the loop means what is that? The free introns after the removal of the intron from the mature RNA. What happened? Lariat with the sloop or a tail, which completely rapidly degraded with the help of the enzyme is called as nucleated by nucleases. This process also requires ATP molecule. Okay, my dear. So remaining things we will see the next classes, my dear. Thank you so much for the participation. Take care. Bye. Good night. Thank you, Ajay. Thank you, ma'am. Thank you, ma'am.
Thank you, sir.
Thank you, ma'am.